Hi everyone, this is Kumar, Associate Professor, SNS College of Pharmacy and Health Sciences, belongs to Pharmaceutics Department. Today we are going to discuss the important and very advanced things recently innovated in the genetic engineering, which is widely used nowadays for the research purpose. That is nothing but a CRISPR and Cas9 system. First of all, what is CRISPR? The abbreviation of CRISPR is nothing but clustered, regularly interspaced, short palindromic repeats. We know the meaning of what is palindrome. The same thing here, base pairs continuously repeated in the palindromic manner. So what is Cas9? CRISPR is a system. Cas9 is a associated protein, comes under the category of 9, an enzyme that cuts DNA in a specific site at a particular level. Next, what it is doing? Bacterial immune system discovered in microbes, and it is adapted for precise genome editing. Here, under the bacterial immune system, CRISPR, they are doing a precise genome editing for the research purpose. And this CRISPR is a revolutionized genetic engineering since 2012 for identifying and doing valuable researches under this CRISPR. Doudna and Charpentier, they shared Nobel Prize. It is one of the simplest, fastest and cheapest gene editing so far. Next, what is the role of CRISPR system in bacteria? With one example we are going to discuss. Apart from this, there are so many roles are there. Next, it's act as a adaptive immune defense against bacteriophages. Is nothing but a virus that affecting the bacteria. Here, bacteria capture viral DNA snippets. It is nothing but once the virus affect the bacteria, it's try to mixing its genome with the bacterial genome. Here, CRISPR array stores spaced manner when the bacteria captures the viral genome, integrate its genome towards the bacterial body. This is the remembering system. Next, when the virus attacks again, CRISPR induced genome editing, the RNA, that is nothing but a CRISPR RNA, guide the Cas9 protein system to match the viral DNA. Next, after matching the viral DNA, Cas9 cuts the invading viral DNA. That's why virus replication or bacteriophages replication is prevented or destroyed inside the bacterial body. Next, it completely fits in their memory system. So whenever there is a situation, bacteria remember and fight the repeat infections. Next, what is the mechanism? How the CRISPR-Cas9 combination works? There are two types of RNA, cRNA and transfer RNA. It is nothing but a combination of these two is called as a single guide RNA. The two essential non-coding RNA molecules in the bacterial CRISPR-Cas9 system that combine to form a functional guide RNA. It's very important. It's a gRNA, and it directs the Cas9 enzyme to specific DNA targets for the gene. Next, gRNA directs Cas9 to exact target DNA sequence, approximately 20 nucleotides matching manner. Next very important thing, we need a specific recognition site for this CRISPR-Cas9 mechanism. That specific recognition site is nothing but a PAM site. PAM is nothing but a protospacer adjacent motif. It is a small sequence in a CRISPR, and typically it's a 2 to 6 base pairs around. It is located next to the target DNA, that means nearby the Cas9 protein, and it recognized to bind and cut the DNA sequence, that is a next gen to target for the recognition site. So what is the main role? Cas9 cuts both DNA strands, creates double strand break. It's nothing but a DSB, double strand breaks. Next, the very important role is cell repairs break. It leads to gene knockout or editing. If you want to remove the gene or if you want to edit the gene, here the combination of the CRISPR and Cas9 plays a vital role, especially in the plants. It plays a vital role, and there are so many excellent researches, gene editing and gene knockout are going on under the plant level research. Next, DNA repair pathways after cut. Here there are two types of things when DNA repair pathways after cutting. One is NHEJ, another one is the HDR. NHEJ is nothing but a non-homologous end joining, and HDR is nothing but a homology directed repair. So what is the role of these two things? So NHEJ is nothing but often creates a small insertions, deletions, and especially for the gene knockout and disruption. And HDR is nothing but a precise repair system, and they need a donor DNA template. So they both allows gene correction, insertion or replacement under the CRISPR-Cas9 system. Especially among these two, NHEJ, non-homologous end joining, is dominant in most cells when compared to the HDR. Next, what are the key applications and future potential directions? So it corrects, especially CRISPR-Cas9 plays a vital role in the plant cells. It corrects genetic diseases. Example, in mammalians, sickle cell, muscular dystrophy, and useful in the cancer therapies to identify the cancer expressing gene to remove it. In agriculture, it plays a vital role to identify the disease resistant crops, higher yield, and better nutrition. Next, what are the research we can do? Gene knockout studies and identify the functional gene and its discoveries and suitable animal models. So there are some other things like diagnostic purpose, antiviral therapies, and used in the synthetic biology. So what are the challenges remain under this CRISPR-Cas9 system? So the way delivery to the CRISPR-Cas9, it's a bit complicated. So with a proper trained person only it is possible. Next, there are so many ethical concerns, especially for the germline editing. So it is varies from one country to another country. So we can't apply this CRISPR-Cas9 for the germline editing without the proper approval.